First comment, we have two Matts on the panel. Two, I used to have their hair. They have a very sad future ahead for hair. I'm just saying, they look great now. This is a talk about what we do, and it's by me and Hanan, who's been clicking around here, and let's see if we can go through it. There's lots of things that have already, she's normally socially distancing from me. Students socially distance from me because when they get too close, I make them do extra work. It's just terrible. We work on a variety of different animals that drop their metabolic rate. We work on frozen animals. People mention turtles in anoxia, and on a bunch of hibernators. We used to work always on cold hibernators. You've heard a lot about cold hibernators, and now we work on warm hibernators. But there's hot animals in the desert that turn their metabolic rate down to 0 as well. So it's an interesting phenomena for me, and I look at metabolic rates, the metabolism, how they change cells and tissues. When you look at mammals themselves, you find that there's relics in every group of mammals that have stuck with the old way. I believe it's the old way. I believe that the stem mammal, which probably is somewhat close to a tenrec, could drop its metabolic rate because we come from lizards that can drop their metabolic rate. They'll drop the metabolic rate in the cold, lizards will drop them in the heat, and then we developed on further and further. There's some differences in the way animals hibernate. Some have that beautiful pattern of Sandy Martin, but tenrecs themselves just go to sleep for long periods of time, like month after month after month. So there's some variation, but in mammalia, we have animals that can hibernate, and we try to study those to see if there's similarities or differences. In our cold mammals, you can see papers. I'll show you some review articles. They're all the same. I mean, bats haven't been ground squirrels for a hell of a long time, but they do exactly the same things metabolically. They use the same stress kinases, they turn on and off the same genes, they regulate the same tissues in the same way, all down, all down. We then move to a marsupial. These are marsupials and very adorable. And they hibernate in the cold. They actually hibernate in the warm too, but we study, the papers are from the cold. Is a marsupial going to hibernate like a placental, or is this something that has diverged over time? Spoiler alert, yeah, marsupials are the same, but adorable. This is the picture of Sandra Martin. This is from the NIH where we get the tissues. We take animals warm, cold, out of hibernation, into hibernation, and we study all the various aspects. This is a modification of a picture you've already seen as well. This is when I worked with Heldmeyer, and animals slow down. So first they turn off their metabolism, and then they cool down. They cool down like a brick. A bat is the smallest brick, and they cool down like with Q10s that are unthinkable. And then you get to bigger and bigger bricks, and by the time we work on bears, they hardly cool down at all. This is an extremely big brick, but they turned off their metabolism. So I'll try to show you animals hibernating in the warm and in the cold, and they turn off their metabolism. That's the key thing for me. And then I look at reversal and arousal and stuff. These are some cold hibernations. I would suggest that you go to the website from the first slide. All the papers are there and all the talks are there. So this talk is there. You don't have to remember or memorize anything. So there's a whole bunch of cold hibernation ideas that are forming around that. These are our papers, but they come from a number of different labs. And then there's warm torpor. This is something that has caught on in the medical field. When you talk to clinicians, they would rather not cool down humans and cool down human organs. My whole deal is to try to do organ transplant. They would rather leave them warm, and there's animals that torpor in the warm. This is a type of primate. This is a lemur. It torpors in the warm because it lives in Madagascar and it never gets cold. It gets wintry, but wintry means dry, and dry means they have no food, and no food means you starve them. They starve themselves like the white mice do. So when you look at those new white mice studies, they take away all of their food, and even a white mouse gets the point and goes into torpor. This is a bigger animal. We work with a French group, a very large French group, again with the European Space Agency. These are bears, and they hibernate in the cold, but if you take your finger and put it up their little bums, you will find they are very warm. They don't drop their metabolic rate like other animals do. If you do that, they arouse extremely quickly and kill you. So we send grad students out to get the bear tissues, then we study the tissues. I'm way too old to outrun a bear. So the principles of hibernation for me is we always see a metabolic rate reduction. You can see this calorimetrically. You can see this by direct and non-direct means. The outdoor temperature is what it is, but it's not critical. These animals don't slow down because they're cold. And we see most genes and processes turn off. There are some genes that are turned back on for antioxidant defense or to make shock proteins. And you can see the papers there. I've got lists of papers about them. We're focusing now on epigenetics and microRNA control. Epigenetics are where you change the DNA so the transcription factors don't bind, turn everything on, off, off, off. So we find it in all metabolic rate depression situations, and I'll just go over the evidence for that. When you see metabolic rate reduction, one of the first things you see is protein kinases turning on and off. So 3% at least of your genome are protein kinases and phosphatases. There's hundreds and hundreds and hundreds of them. Most of them are not involved. I thought most of them would be turned off, but I don't think so. I think they're just not involved. They have no substrates for the enzyme. These are enzymes that phosphorylate other enzymes, but some turn on, and these are signaling pathways that work from the cell plasma membrane all the way in and then reach into the nucleus, and they reach into the mitochondria. So we do a lot of mitochondrial things too. So all metabolic rates have these things. In theory, if I had enough small children, I could activate these pathways and see if we could hibernate a small child. If you look at a picture of a cell, this is a picture of a cell drawn by a grade 8 student, so don't judge. There's a turning on of some kinases, just literally less than a dozen out of 750, and they reach out into the cell, helping to turn things off. They are semi-involved in epigenetics as well. So the reason I'm mentioning this is there are hundreds of papers. You'll have to read them and memorize them. But really, this is part of epigenetics. So a few SAP kinases, stress kinases are activated, and processes get turned off and off and off and off. And this is why you don't have animals generating heat. Step after step after step, transcription steps, translation steps, they stop breaking down fuels. They switch over to fats, but even fats are mobilized less quickly than if they were at 37 degrees running around. And there's also gene activation. So in a slight change, I had to realize that message RNAs were not being made actively. Messages that were made were not being extended actively. And I found messages in stress granules and P-bodies. I found messages tied up in prison in hibernating cells, prison, and they couldn't get out. They couldn't interact with polysomes. Polysomes were taking a nap. Polysomes were going down to monosomes and not being activated. You also see this in starved mammals that aren't hibernating. You also see this in cell lines. So in cell lines, where you starve the cell lines and don't give it enough fuel, you find this rise in P-bodies. A few genes are activated. We won't focus on them here. So epigenetics for me was something that my cells and animals needed. I needed everything turned off, and I needed a general mechanism other than just some stress kinases to do it. It's a thing. It didn't used to be a thing. I'm so old that there were no epigenetics, that Lamarck was terribly wrong and Darwin was spinning in his grave. We now know some of the mechanisms, and it's an active growing field. If you drew one of those PubMed, it was wonderful. We saw a PubMed thing and numbers of papers per year. Holy God, this is really important that I'm just a tiny little creature who focuses on the hibernation part. There's DNA changes. These DNA changes are so transcription factors can't find DNA. DNA is balled up, DNA is covered up, boom, less. There's histone modifications. You could spend your whole life on histone modifications. I can't even show you all the pictures. We focus on histone 3. We focus on post-translational modifications with lysines and phosphorylations. And there's the regulatory non-coding RNAs, the microRNAs I told you about, which are incredibly exciting. So the paradigm here is you cannot do what you used to do. There's no use pretending. And it's your brain, your liver, your kidney, your heart, your muscle, everything. ATP expensive processes go down. And so when you do calorimetry on these guys, they are taking a dive. And you can change the methylation of genes to reduce transcription rates in promoter regions, in intron-exon overlaps, it's crazy. The methylations happen. The way that modern people do this, and we've done it in some animals, is to do a complete genome, then a complete methyl genome, and then let the machines grind through it. Oh, it's like the old Transformer movies with Skynet. There's histone modifications too many to talk about. I'll show you a couple of papers, and then the microRNAs. So what's happening is these three great changes are involved. You can also see papers of us with other post-transcriptional modifications, long chain RNAs, circular RNAs, and RNA binding proteins. Dr. Martin mentioned RNA binding proteins, so important, yeah, part of the talk. So these common mechanisms probably occur in every animal we've looked at endlessly, in every tissue. These things happen, and we think that they happen in a coordinated fashion because we see them everywhere. So I can't cover all the data, but you can see the papers. You can see them on the website. You can see them in PubMed. DNA methylation happens. We were shocked to begin with. We looked at individual genes. We looked at where methylations were in CPG islands, and then finally we found them everywhere. We found them in insects. We found them in frozen frogs. We found them in sea cucumbers. When you work with BGI, you have to work with sea cucumbers. I don't know what it is. By the way, they taste awful. And it eventually happened in hibernators. We had to work with experts on hibernators, and now we do genomes and methyl genomes to see what happens. So there's lots of papers from our lab. You can see them, and then you can look at the references to the other papers in other animals. Histone modifications happen. Like I said, we focus on histone 3. We don't measure all of the impacts, but these are a set of papers that come out, and we've got a bunch in 2020. Of course, you can't sort of stop these sort of studies. You have to continue on. You find more and more. It's one of those things where you find 2 or 3, and then it's like tumbling down with down a rabbit hole like Alice in Wonderland. But it's amazing. These are really interesting phenomena that change. The principle of all of these things, the principles of summarizing 150 papers, is metabolic pathways do produce small molecular weight molecules. Metabolic pathways aren't shut off completely. These small molecular weight molecules, then I'm metabolism, boom, I'm going to the nucleus. I'm going to deal with kinases and methylases and demethylases there. They're called writers and readers and what's the other one? Writers and readers and erasers. So it's way too complicated for me. I'm just a guy. I work on some of these things, but it's the integration of metabolism. We're not doing that anymore. We're doing less of that with, okay, let's turn off the genome as well. Let's take a nap. MicroRNAs are where we're focusing our interests now. My interests are really my students' interests. Hanan, our best student ever, is interested in microRNA, and therefore I am as well. So they've driven me to this. MicroRNAs used to be really interesting for me to do, and now it's just like way complicated and involves machines and computers. Initially, we tried to look at 1 or 2 microRNAs at a time to see what happened, and that really worked. But now you have to look at them all. So there's way many of them, and you do an ohm. So you'll see in our papers, we do proteomes and phosphoproteomes and genomes and metabolomes. Those I hate the worst. But this is microRNAs. They're understandable. Once you look at them, you find that microRNAs are really, really conserved. If I homogenized you and I homogenized a cow, I don't think we could tell the difference by looking at your microRNAs. They stay the same. They're in all mammals all the time. We can use some of the same tools in all the mammals. MicroRNAs of a given type have a given function, and you can actually extrapolate those functions across mammals, but there's also mammalian-specific ones. There's specific ones for your species. We used to call them unknown microRNAs, and then we realized, dude, we know them. So now we have to call them species-specific microRNAs. I think they're a key to disease. I think that after my death, when you young people are still alive and hobbling into your doctor, he will have a microRNA array, and he'll be able to tell what cancer you have, where the tumor is, and what stage you're in, just from the changes, these waves of changes of microRNA. It's amazing stuff. It's coded for in your genes. It's made as a product, but it's never going to make a protein. This isn't message hiding. This isn't a long chain that's been truncated. This is made to be a microRNA, and there may be 1,000 of them. It's then polished and sent out. We study the pathway. You can see Droscher and Dyson, Argo. So we study the pathway, and in hibernation, where metabolism turns off, microRNAs go up, and this pathway is strengthened. So you can see tissues taking to hibernation that are turning on the making of microRNAs, that are kicking microRNAs out, and it's always bad for processes, right? They can bind to message RNAs. They can destroy message RNAs. We don't see much of that. They turn things off. They go up. They turn things down, exactly what a hibernator would like, exactly what you'd like if you were preparing an organ for transplant. You take an organ out, you just microRNA the very dickens out of it, and then it would turn off its own metabolism. So we study this a little bit with a group in UCLA who does organ transplants, and a little bit with a group in Chicago that does monkeys. So we don't talk about the monkeys. Oh, look at this. So here's what happens. There's too many papers, but they're all exciting. You have to go to PubMed. Just go to PubMed, just sign me into PubMed and look at them. You only have to read the abstracts, but they're tremendously exciting. I want to sell them. We used to do a couple, now you have to do them all. So what we do is we do an ohm, measure them all, but then the data that comes back is from machines. So you got to modify and you got to cut it up and you got to put it into tables and you got to figure out what's going on. So in that way, it's like a transcriptome, but these are predictive. If I see 4 microRNAs, working together and I know what they do, from a human atlas, boom, I can tell you what's off. So I can tell you what's off. These actually are control elements, not just downstream events that may or may not be important to hibernation. Marsupials, yeah, they went along with the program. So at some point, our turtles, our lizards, our frogs, they have microRNA as well, and they're very conserved, and they turn them on and off with metabolic rate changes. Marsupials, mammals, we all bought into the ancient ways of the reptiles, and they have very similar processes, very similar microRNAs, very similar processes, and therefore when you look at their biochemistry, it looks the same as well. That's because the microRNA are directing the show. They're like a conductor of an orchestra with all the metabolic pathways I used to love, parts of the orchestra. So now we have the possibility of having a warm primate. Now, you might want to send a warm hibernating primate to space. And if you send lemurs, they're really, really smart and they have opposable thumbs, dude, they can probably do the job. On the other hand, lots of people want to send bears because when you send a bear, when they get off at the other planet, they rule. So you'll have to decide what's best for space travel. These are a warm animal that drop their metabolic rate, but not their temperature. Dr. Martin mentioned this as a phenomenon as well. So it's food regulated. There's actually 9 different trophic zones in Madagascar. It's a nightmare. We now work on tenrecs from Madagascar, and they differ in their different zones. You know lemurs as the big ones. Remember the big lemurs with the striped tails? Those are bullies and idiots. Number 1, they don't hibernate. Number 2, they live where it's warm and wet all the time. My little guys have to live on the edges where the trade winds mean that 6 months of the year they have no food and are viciously preyed on by these huge, scary raptors. So we wanted to know if this is the same. Are these like totally different? Or is this just a variation on a theme? When you look at their body temperature and the air temperature, you find it's quite warm. That initial diagram was for cold. When you look at their metabolic rate, however, it goes way down. You can see the metabolic rate there. So we studied them all, and you really don't want to know all the details. But there's a series of papers on lemurs, and there's more now because you have to do microRNA and you have to do other things. You have to do genetics. What happened was they have very few changes. Compared to the ground squirrel, compared to all the complex things you saw by really good labs on animals that cool down and you're cold and you're hibernating, these guys don't do that. They don't do shock proteins. They don't do all of the stress pathways. They essentially have invested in microRNAs, and these microRNAs turn things down where your metabolic rate goes down. But a lot of the changes you see for ground squirrels are because they're cold. And the microRNAs here are really good, and you can look at them and learn something. Can't talk about all the microRNAs, but the hibernating primate is now a source of, hey, can we take this in human cells? Can we take this in heart cells from transplant patients and try to turn those off? And although I'm not allowed to mention it, what about monkey arms? Oh God, don't ask. But here's an example of a pathway that we didn't know existed, but you can see it from pathway studies, WNT, and boom, microRNA is regulated. So a brand new pathway shows up and you go, how exciting? Oh yeah, microRNA. So microRNA is the answer. So no matter what your question is, microRNAs are the answer. And you can add them to tissues and cells. You can add them to organs. So if you're looking to shut down, if you're looking to shut down something going, keep it warm, identify key processes, and then just soak them with microRNAs as much as you can until you're tired. All right. Yay! Again, I have very bright people who work with me. I have wonderful colleagues in the US, Dr. Hallenbach and others. And I'm a product of my support staff. So, am I good? Did I finish on time? Is there time for a question?
Yeah, thank you very much, Dr. Storey. We have close to 6 minutes for questions. So great, thank you very much. It was really a great presentation.
Who here thinks that if I knew anything more, I wouldn't have told you in the talk? Who? Okay, all right.
There's always a question and a most entertaining talk. Thank you very much. First question here is, RNAi compounds are in development by the pharmaceutical industry to modify gene expression for certain proteins. For example, there's RNAi that can shut off the production of protein that regulates cholesterol metabolism. Could specific RNAis be developed to selectively shut off, say, bone resorption during a long duration spaceflight without necessarily having to induce torpor, or as well as could that approach be used for initiating the torpor-like states?
Oh, absolutely. There's no question microRNA is good. And RNAi is great because you can feed it to Drosophila. We feed it to Drosophila. They eat it and it acts within them. You can pour it onto cells in a dish and they'll take it up. So yeah, RNA is the example. But here's what you missed in my talk. I don't do experiments. I walk the earth. I find things living under rocks or at the top of trees. So if I have a choice of going to the top of Mauna Kea and finding insects that are freeze tolerant, I'll do that instead of developing, what did you say? Developing tools to, no, I don't do experiments. So you guys have to do experiments, but just be sure to reference me, unlike other people in the hibernation field.
Very good, thank you. Marmosets express torpor, why not study them?
Because we can't get a hold of them. So here's the thing. If I can get a hold of an animal, I can get a hold of an animal. Like animals at the top of mountains in Chile are not safe from me. We study squid that live at the bottom of the ocean, and they thought all they had to worry about were whales coming down and eating them. Yes, marmosets hibernate. They live in Brazil, but with COVID, I can't get to Brazil. They can't get to here. I know people in Colombia. If I live long enough, I will know everything about marmosets. And all you have to do is take my lemur papers, cross out the word lemur, and put in the word marmoset, baby, and we're good to go. Yes, there are other models, but I can't study everything.
That's fair. Next question is microRNA the answer to why we have failed so far to factor out infradian cycles and other Zeitgebers?
I believe so. So here's the thing. You had a war on cancer in the 1970s, and we knew so little that well-meaning people thought well, we're going to get cancer if we put in enough time. But there were so many unknowns that we didn't know. Boom. And you can see it in COVID as well. Well, now that we know it's spread on surfaces, we could, no, it's not spread on surfaces. This is a brand new paradigm. This is a brand new way of looking at things. This is a window into a construction zone that we didn't know existed. Yes. No matter what your question is, microRNA is the answer. Wait, I already predicted that, didn't I? Oh, God, I am good.
That makes it easy, although this question is from me personally, and I'm not sure you can answer it that way. What relationship do you see, if any, between the adenosine findings from our first earlier speakers today and the epigenetic, histone, and microRNA findings that you're describing?
Adenosine is part of a group of nucleotides. So you have ATP, fish have ATP. When you exercise, your ATP levels go down. When fish exercise, they change ATP into IMP and keep their energy charge high. If you look at adenosine as part of a dozen different molecules that are related to each other, that all interact, and that I published papers on, go back to the 1980s, yes, I believe that epigenetics is going to regulate the enzymes and enzyme activities and amounts of the enzymes that interact inosine, adenosine, adenosine monophosphate, GMP. Yeah, I think, but that's how it does. Epigenetics cannot reach out and increase or decrease a molecule, but they can reach into the process that makes or breaks a molecule, boom, and turn it right off. So yeah, I think there's a possibility. It will take a lot of time and energy. It's really difficult. You may have noticed I don't do things that are difficult either. So.
But it does appear that you do things that are fun. So that's important in life too.
Oh yeah, I think that if here's the problem with brain studies. Oh, this is an area of the brain. Come on, there's 9 different cell types in there, and the cell types are interacting on a level that you can't possibly figure out. So I think that at some point, bright young people will have single cell extraction techniques. We've tried them in some cases, and then they can look at individual cells. Now, mole rats have been taken over by evil corporations. Oh, yeah, it's Calico, it's Buffy. And they're now doing single cell phosphoproteomics, single cell microRNAomics. And so at some point, if there's enough energy, enough time, enough money, yeah, we will eventually be able to sort these things out. The people that work on these problems are very brave. We have a word in biochemistry, it's called brave. It means crazy, it's never going to work, and it just takes too much grant money. So I believe that all the studies you've heard are very brave.
Okay, last question here. Do microRNAs become exosomes when they are packaged?
Yes, yes, they do. They, okay, Dr. Martin mentioned a snapshot. I'm giving you a snapshot. I'm giving you the ultimate snapshot. Dude, we whacked this guy, we measured it. They are made in cells as precursors, then they're activated and sent out. Exosomes are full of microRNAs from a tissue, but we don't know what tissue. And we don't know the concentration of any macromolecule is equal to the rate of its production minus the rate of its degradation. And here you got blood carrying stuff around. Exosomes are going to be a key thing for cancer and other diagnoses. They're going to take your blood, they're going to just whack it down, and they're going to tell you what there is. I think cancers, oh, we got rid of all the cells, and you're free of cancer. Oh, it's back. It's back from a mixture of microRNAs and messenger RNAs that have been carried around in a ball that you can't even see. They wait for a while, and then they hit a cell. Too many unknowns. If you study kinetics at all, there is just too many variables. You can't do any sort of Cleveland kinetics on this. There's just too many unknowns.
Excellent. Well, thank you for all of your answers. And with that, I think that puts us right on time for those who need a bio break and food.
Yeah, thank you very much, Dr. Storey and Dr. Strangman. We have a 30-minute break until 1 p.m. Central. We'll come back for our last 2 speakers, and then we will have our panel. So we'll see you, everybody, at 1 o'clock Central. Thank you.